Thank you. It's a pleasure to be here, and it's especially nice to be invited by students, which is definitely an invitation you do not want to decline. So it's, again, thanks very much for the invitation. It's been great seeing lots of old friends, meeting some new people, and I've enjoyed my visit so far. We'll see what happens during the questions, right? Okay. So this is a pandemic snapshot from my iPhone that I took when San Francisco was completely deserted and I could bike anywhere without worrying about traffic. And this is from the top of Twin Peaks, and it's just a little reminder of where we're at. And the traffic and the tourists are back, which I guess is a good thing, but I do miss a little bit being able to just be free in San Francisco. So since this is a genetics colloquium, and I view myself first and foremost as a geneticist, many of you will be familiar with this term that is, let's see, I'm not advancing. That's not good. Do I have to click on something? There we go. Okay. Something called the awesome power of yeast genetics, which some people say was coined by Ira Herskowitz, one of my former colleagues, and heavily used by Christine Guthrie, who happened to be my thesis advisor. Christine passed away last summer, and it was nice to write a piece and obituary with David Brow here, who was briefly a lab mate of mine back in the day. So the awesome power of yeast genetics means a lot, but at some level, it's a certain aesthetic of how you approach science using genetics. It's sort of hard to replicate in any other system for a variety of reasons, mostly technical. And so today what I want to talk about is our attempts to bring the awesome power of yeast genetics to human cells. This is Irene Busch, who's a postdoc in the lab, who came from a structural biology lab in Switzerland, Fred Allen's lab, interviewed with me and said, I want to join your lab. And I said, great. And she said, but I will only join if you let me work on human cells and on RNA. And I said, okay. And so whenever I told people this early in her time in the lab, people said, you said yes? So, and for this project, Irene got help from two technicians that she trained, Beidou Rao and Susan Lee. Beidou was poached by Dan Goshling at Palico, where she now makes double her previous salary. And Susan Lee is a grad student at UC Davis. And this is at our lab, one of our pandemic lab holiday gift exchanges. So a little cell phone video. Okay, so yeast genetics, human cells, what's the question? So Irene wanted to study RNA, and in particular RNA splicing. So I'm going to give an introduction to those of you who do not work on RNA splicing, and there's a few of you in the audience. So as many of you know, genes in humans mostly encode introns, and introns tend to be large relative to exons in human cells. They vary dramatically in size, some as small as less than 50 nucleotides, some as large as over 110,000 base pairs. Many of you will be familiar with splicing as being a simple chemical reaction involving two sequential transesterification reactions in which a nucleophile within the intron in a branch point adenosine, a 2' hydroxyl, attacks the phosphate at the 5' splice site, resulting in the production of a cleaved 5' exon in this famous layered intermediate species, which is then further processed through the attack of the 3' hydroxyl of the cleaved 5' exon on the phosphate at the 3' splice site, leading to ligated exons and the excision of the intron as a lariat, which is then debranched and degraded. The enzyme that mediates this reaction is a ribozyme, and it functions by holding precisely two metal ions that stabilize the transition state of both the first and second chemical steps of the reaction. And this enzyme is interesting, not just because it's a ribozyme, but because this ribozyme is formed by a massive RNA rearrangement, which is really unprecedented anywhere else in biology to make an active enzyme. And so the fundamental question then becomes, how do you choose the right substrates? How are the correct phosphates chosen for the cleavage ligation reactions of this enzyme? There are a number of problems that need to be solved here, because imagine an intron with 110,000 nucleotides. How do you decide which phosphates to place in the active sites at the right time, and in the right phosphates and at the right time? So this is a problem particularly from organisms like humans that have very little information content around the actual sites of cleavage and ligation. So the 5' splice site is basically in RNA, a GU, the branch point is usually an adenosine, and the 3' splice site is usually a pyrimidine AG sequence. There's a little more information upstream in a polypyrimidine tract. Contrast this to Cerevisiae, which people have referred to as a hardwired species, in which the sequences that define introns are highly conserved across the small number of introns that this organism harbors. Some of this is, of course, solved by the action of sequence-specific RNA binding proteins that recognize specific sequences in the flanking regions that help position the factors that recognize these sequences during assembly of the enzyme. A second problem is the fact that there are multiple introns in genes typically. The record holder is Titin, which you all need for your heart to pump right now. So the Titin gene encodes 362 introns that have to be spliced correctly for this protein to be made. And so a fundamental question that, again, is only, I think, partially answered is how do you avoid skipping an exon? And this is really a problem when you consider the fact that sometimes you want to skip an exon when you have a process of alternative splicing where you need to make many isoforms from a single primary transcript. The vast majority of human primary transcripts are alternatively processed through a combination of alternative promoters, cleavage polyadenylation sites, and splicing. The record holder, though, comes from Drosophila, in which the DS-CAM protein, which is involved in neurite identity sensing, literally has 38,000 different isoforms that can be made. And each of these proteins, particularly those with different extracellular domains, has a different binding specificity, which is still one of the most remarkable genes and proteins ever identified. And again, one transcript, one gene, that many different proteins. Finally, by way of introduction, I want to just briefly mention that splicing plays important roles in human diseases. There are many examples of this, but one I want to mention for reasons that will become clear is that one surprise of the Cancer Genome Atlas project was the identification in some tumors that have at a very high frequency mutations in this essential machinery that mediates the splicing of introns, and that's the spliceosome. So in a myeloid tumor called myelodysplasia, which is basically a pre-neoplastic state that converts to leukemia, 60% of the tumors harbor mutations in spliceosomes. And this is just the original paper from a group in Japan, and they affect a variety of different proteins that are mostly involved in early recognition events. And it's thought that these mutations cause alterations in the splicing of key downstream factors that control the differentiation state of myeloid cells. I'm not going to talk about all of these factors, but I want to mention one factor, which is highlighted here, which is called SF3B1. This protein is frequently mutated in myelodysplasia and leukemia, and it's also mutated in many human cancers at varying frequencies. I'd say on average about 5% of human cancers have mutations in this protein, and some of these others. And what I want to mention right now is that this protein is a large protein with many of these heat repeats, and you can see that there's some hot spots within these heat repeats that are found in tumors. Okay, so because of these, the results of the Cancer Genome Atlas efforts, there's been efforts at drugging the spliceosome to treat these tumors with the idea that maybe if you could find compounds that selectively killed the mutant tumors, you'd have a therapeutic index that allowed you to have an effective treatment. Despite these early hopes, one of the biggest efforts did not succeed in this area, probably for a variety of reasons. But it's fair to say, I think, so far that drugging, spliceosome mutated tumors has yet to succeed clinically. As a basic scientist, I might say that, well, that's because we don't really understand this machine. So major questions remain unanswered relating to not only the core mechanisms, but accuracy of the process, how it's regulated, how it can be plastic, and also really how these mutations cause disease. All of these have major unanswered questions. So for those of you who work on splicing, you know that the spliceosome is incredibly dynamic. So again, the spliceosome is the enzyme that mediates splicing, but it's not one thing. It's a series of protein complexes that assemble through a largely ordered pathway, which involves recognition of consensus sequences at the 5' splice site around the branch point, this polypyrimidine tract that I mentioned, and the 3' splice site. And there's multiple recognition events that happen. There's a initial recognition step. There's an activation of the enzyme through this RNA rearrangement that I mentioned before, followed by the chemical steps and the disassembly of the spliceosome. There's, as I'll show you in a moment, a huge number of factors involved, some of which are small nuclear ribonuclear proteins, or SNRPs, U1, U2, U4, U6, and U5, a number of other protein subcomplexes, and a number of sort of free proteins that assemble at various stages, come on and off during this very complicated process. There are about 8 ATP-dependent steps in the yeast Saccharomyces cerevisiae. There may be more in humans. So this means that this process is really excessively complicated, right? What enzyme that catalyzes 2 chemical steps needs all of this rigmarole, especially when the core enzyme is pretty simple, involves, a ribozyme. And so nobody knows for sure what the answer to this is, but one idea for which there's some evidence is that every one of these steps gives you an opportunity to check whether you're doing the right thing. So the idea is that there are many checkpoints or proofreading steps in this cycle. And the evidence for this comes almost entirely from studies done in yeast, first genetic studies in Christine Guthrie's lab, and then biochemical studies in John Staley's lab at the University of Chicago. And before I get to that, I just want to mention that, yeah, there's a lot of proteins. Human spliceosome has 153 proteins, and mechanistically, the function of many, perhaps most of these, is not really well defined. Okay, but why is it so complex? Why so many ATP-dependent steps? And so the going theory is that it's proofreading, that there are ATP-dependent steps that act as essentially as molecular timers that enable kinetic proofreading. And this particular scheme has been put forth from the yeast work, and it's simplified here. I've stolen this from an old review from John Staley's lab. So imagine you have a substrate that gets converted to a product, but imagine that you couple this a chemical or enzymatic reaction with an ATP-dependent steps, where an ATPase essentially acts as a molecular timer. And the same ATPase is involved whether it's the substrate, whether you're in the substrate state or whether you've been converted to the product state. And so the idea is that if the hydrolysis happens before chemistry has happened, then the substrate requires some kind of rejected state and is discarded or destined for some sort of destruction. Whereas if the chemistry happens and then you have ATP hydrolysis, now that product can continue to be processed through this multi-step pathway. And so this scheme, there's very good evidence from, again, Staley's lab for this scheme at the catalytic stages of spliceosome function or assembly in yeast. And this has all been done using mutant substrates, where you mutate one of those signals and find that those mutant substrates are indeed discarded. And if you slow down the ATP hydrolysis or just prevent the discard step entirely, you can now move through the forward pathway. So the general idea is that all these steps and all this complexity is really there to check to see if things are working right, and if not, to cut bait. And so, again, this has been established in yeast. It's been established from mutant substrates. It's not clear whether it happens at earlier stages and whether it happens in other organisms. Okay, so I know I've thrown a lot of complexity about splicing at you so far. For today's talk, you don't need to know all that stuff, and I'm just going to give you what salient background you really need to know to understand the rest of the talk. And all you need to know are a couple early steps. So the earliest step of spliceosome assembly, at least in sort of the canon, is that U1 recognizes the 5' splice site by Watson-Crick base pairing between U1 snRNA and the 5' splice site consensus sequence. The sequences around the branch point are recognized by a sequence-specific RNA binding protein called SF1, or branch point binding protein. And then the polypyrimidine tract and 3' splice site are recognized by a 2 subunit protein called U2AF that binds to these sequences cooperatively with SF1. And this complex is called the E complex. It's quite floppy, so there's no cryo-EM structure of the whole thing, despite many efforts. This is converted in an ATP-dependent step, and actually it's many steps that are not well worked out, to the A complex, in which these proteins are somehow displaced. U2 is recruited by these proteins, and then somehow they're displaced through a reaction that is very poorly understood to form a U1, U2 complex where these two SNRPs interact with each other, and that's called the A complex. And there's a whole bunch of other complexes that we're not going to talk about today. So that's really all you need to know. But I lied. There's one more thing you need to know. Back to this protein SF3B1 I mentioned before. This is a U2 SNRP protein that's part of the SF3B complex, which is a subcomplex within U2 SNRP. So this is U2 SNRP, even though its function is to just bind the branch point, is a 17S particle. It's very large, complex protein particle, ribonuclear particle, ribonuclear protein particle, because it has an S and RNA. So SF3B1 has these heat repeats that I mentioned before that form this this sort of alpha helix, this alpha solenoid, where you have alpha helices forming a superhelix. There's 20 of these repeats, and it's conformationally dynamic, and it has sort of this, at the extremes, an open conformation, and then clamps down to a closed conformation. And during the splicing process, it's thought that the closed conformation, well, it does clamp down on the U2 branch point helix. So U2 recognizes the branch point by Watson-Crick base pairing, and this protein complex clamps down on it. And that's an obligate step in splicing. Later on during this process, after this clamp is closed, it actually has to be opened and removed. And that occurs later in one of these ATP-dependent steps by an ATPase that's of this DEAH box family that I'll be talking about a little more later on. So again, you need to know that there's early steps involving U1, branch point binding protein, or SF1 and U2AF, ATP-dependent step involving the removal of these proteins and association of U1 and U2, and then later on something called the triple SNRP adds, and the reaction, the assembly process continues. And you have to remember this happens separately for every single messenger RNA precursor. Every time a gene is transcribed, every intron has to go through these recognition mechanisms. Okay, so Irene wanted to study splicing, but we needed an approach. And I said, being a yeast person, it's got to be something close to what you can do in yeast. And of course, there are problems. And the motivation is simple, right? The function of most spliceosomal proteins is unknown. And the known functions have mostly been determined by studies in Cerevisiae, in vivo or in vitro. Human spliceosomes contain many proteins not found, I mean, dozens and dozens not found in Cerevisiae, but you can't really do yeast genetics in human cells. One problem is spliceosomal proteins are essential, so you can't just knock out the gene and figure out what it does. Another problem is that it's hard to make point mutations. Another problem is that human cells, at best, are diploid, and most cell lines are something ploid, triploid, God knows what ploid. Okay, so after brainstorming a lot and talking with a lot of people, we decided to use as a mutagen something called a CRISPR base editor. So this is technology developed in David Liu's lab at Harvard that takes advantage of the fact that if you deaminate cytosine or adenosine, you change the base to a different base that has different base pairing potential. So if you deaminate cytosine to uracil and you don't repair that damage in DNA, you now get a C to T transition. And so what David Liu figured out is that if you take Cas9 and mutate it so it only nicks one of the two DNA strands, so it doesn't make a double strand break, and then you tether to it an engineered single strand specific deaminase, that once Cas9 binds to its target programmed by a guide RNA, it will, within a certain region, deaminate any Cs in that region and produce, again, C to T transition mutants. There's some bells and whistles that have to be generated to make this work, but basically, this method has some efficacy in model systems. So the idea was to use this to mutagenize the entire spliceosome. When we started this project, and even today, CRISPR base editing screens have yet to be applied to an essential cellular machine in human cells. While we were doing these studies, a couple groups described sort of proof of principle screens where they mutagenize, bunches of proteins and stuff, but nothing was done on this sort of epic scale because of a number of technical challenges with the system. One was finding a base editor that you could make a stable cell line with that didn't kill the cells, and another challenge I'll mention in a second. But the general idea was to make a library that covers all of these proteins. So what we did is we designed more than 40,000 guides that targeted every exon of every protein in the spliceosome. We didn't do the small nuclear RNAs in the spliceosome because they're multi-gene families, so we figured that wouldn't work. Maybe we should have. And so basically we have guides against every possible site and every coding exon of all of these proteins. And that gives you a smattering of Cs that can be changed to Ts. One can computationally try to predict how much mutagenesis this is. And one guesses basically that about 10% of nucleotides can be changed by this methodology. And about half of those substitutions will produce missense mutations, and then some even worse. And so that's the library we made, but we needed something like a yeast cell that was haploid that we could do genetics with. And so the only game in town are EHAPs, or this is a derivative of a cell line called HAP1, which is a derivative of another cell line called KBM7. KBM7 is a chronic myelogenous leukemia cell line. It's Philadelphia chromosome positive. And the original KBM7 was almost just by chance, weird cancer line haploid. EHAPs have been engineered to be completely haploid. There was still a diploid region of the genome. So this is crucial because it enables one to connect genotype and phenotype in pooled screens. I will say that these cells are a pain. Any of you who've worked with these are a pain to deal with because if you let them get confluent and stress them, they become diploid, and the diploids take over the population. So one has to, work at keeping them happy and keeping them haploid. One trick we use with is that there's a compound that was described in the literature while we were doing this work that selectively kills the diploids. It's an expensive compound, so we don't use it every day. But it took a lot of time to derive a stable cell line that expressed an effective base editor stably and remained haploid and was happy. But once we got it, we froze it away. Okay. So what do we do now that we think we can mutagenize a cell in a sort of yeast-like way? And so the initial perturbation we chose, and this was just sort of to do a proof of principle experiment, was using a small molecule, a compound called plidenylide B, which is a U2 SNRP inhibitor. So what I'm showing you here, once again, is that SF3B complex. And it just happens that really the only sort of high affinity lethal compounds that block splicing bind in a little pocket right here. Plidenylide B is one of them that's commercially available, and it binds in a little pocket between that protein I've already mentioned twice, SF3B1, and PHF5A, which are subunits of this SF3B complex. And they're crystal structures, and there's a lot understood about this reaction. And this kills cells dead, and it blocks spliceosome assembly. You can make something like an A complex, but you don't go any further. So the proof of principle experiment was to ask, well, if we mutagenize cells with this base editor library, and then we screen for resistance mutants in these haploid cells, can we get a viable mutation in the cell line that gives you resistance to these drugs? And do they make sense where they are? And remember, we're now mutagenizing 42,000 sites. And so the experiment is a very standard CRISPR screen where you transduce your cell line with a guide library, you select for the transductants, and then you collect cells at an initial time point, and you split into plus and minus drug, wait 2 weeks, and then you sequence guides and analyze the data. Importantly, Irene was able to figure out, was a good farmer of HAP cells, and so she could keep them haploid during the course of the mutagenesis. So if you don't give them drug, they stay haploid through the process. Drug stresses them out, so you start to get some diploids, but we figure it doesn't matter as long as while you were making the mutants, they were haploid. Okay, so we did the screen, and what I'm going to show you is a plot with lots of dots on it, which I'll explain carefully. So every dot in this plot represents the a single guide RNA, okay? And so there's, 42,000 points or whatever here. So on the x-axis is the read count across all the samples, and on the y-axis is the ratio of guide abundance with drug to without drug on a log scale. So what you see are dots above the line and dots below the line. The dots above the line represents resistance, and the dots below the line represents hypersensitivity. We did a very simple, we used what everyone uses for RNA-seq analysis, DE-seq2, to pick significantly over or underrepresented guides. And so what you can see is that very nicely, we got guides and proteins that we expected to, PHF5A and SF3B1. I'll tell you a little bit more about them in a second, but we were happy to see that in this sort of simple proof of principle experiment, because experiments like this have 42,000 ways of going wrong. What we didn't expect, but should have, was guides that produce a hypersensitive phenotype. So this is the equivalent of synthetic lethality, and they actually made some sense. But before we did anything with these, we wanted to confirm the data, so we validated the results in these competition experiments where we use individual guides. We mark the cell lines with fluorescent proteins, and we either use a non-targeting guide or targeting guide. We transduce the cells, and then we mix them 1-to-1. And the targeting guide is labeled with a different fluorescent protein. And then we compete them against each other, the non-targeting versus the targeting. And then we use flow cytometry after 2 weeks to see who won. And if we compete two non-targeting guides to each other, the triangles are with drug and the circles are without drug, nobody wins because we didn't change the competition. But if we use guides that, for example, produce hypersensitivity to drug, or at least predicted to based on our initial analysis, you can see that the circles go down over time relative to the triangles, meaning that indeed these guides program a sensitivity phenotype, and guides that promote resistance give you a winner in the competition in the presence of drug. So, and basically our very simple bioinformatic approach worked really well, and every single one of the guides up there that DE-seq2 picked as being significant retested in these independent validation experiments. For the hypersensitive mutants, we were happy to see that most of them ended up in U2 SNRP, since this compound inhibits the activity of U2 SNRP. It was perhaps gratifying to see that there were mutants in U2 SNRP, point mutants in these proteins. And again, these are viable point mutants. The cells have to be able to grow, that are hypersensitive to this small molecule, as well as mutations in a number of other factors that we're still scratching our heads about a little bit, that also give you hypersensitivity to the compound. We can, as I'll mention in a moment, figure out what the mutations are and map them onto all the cryo-EM structures that we now have in the spliceosome, including human spliceosome. So here's one example where this is a structure that has the drug, or the compound, I should call it a compound, not a drug, the compound bound. And you can see there's a region over here that produces hypersensitivity to the compound. Mechanistically, we don't know what's going on, but if you zoom out, that's that little patch there. That's where the drug binds. We can also find mutations in other components over here. And we suspect it affects this ability of this sort of horseshoe-like clamp to open and close, and that dynamics is somehow being impacted by these mutations. But it will require some biochemistry to really test those sorts of ideas. But it's fun to be able to map the mutations onto structures. What I haven't told you is how you figure out what the mutations are, and it's a little trickier than you might expect. So what we do is we transduce the cells once again independently with the guide, and then wait until the mutations have happened, and then split again the cells into plus and minus compound arms, extract genomic DNA, and then we amplify the region around the guide target site, and then we do Illumina sequencing on this library for each and every guide that we got out of original screen. And so the reason that's, so the way that works is that you end up with reads that have substitutions in them, and you can count them, which gives you the prevalence of each predicted protein sequence. Of course, you can translate the sequence and get the predicted amino acid changes. And then you can watch what happens to the abundance of the guides of each of these mutants, basically, over time, with and without drug, and then make conclusions about what is the causal mutation. So in some cases, this sort of analysis is fairly straightforward. So here's a guide against SF3B that produces resistance to the compound. So we know this guide produces resistance. What does it do to the DNA? On the top row here is the wild type sequence, and shown over here is the abundance of the wild type sequence. And the second row is this particular amino acid change, threonine 1080 to isoleucine. And what you can see is that after transduction, there's barely any abundance of this sequence. So it may be that this is just a very bad guide in terms of its efficiency, or it could be that it's toxic, we don't know. But what you can see is that when you add, so in the presence of the solvent DMSO, this guide is not enriched, this sequence rather is not enriched, but in the presence of pladaenolide B, now this, the sequence encoding this amino acid change takes over the population, as shown here, whereas, and then wild type goes down in abundance over time. And then you can also plot this as a log fold change. So if you don't like reading this, you can just look at the colors here. So this is how one uses amplicon sequencing to figure out what mutations are being enriched. The reason this is important is that base editors don't always do what they're supposed to do. So here's an example of a guide against the other protein that sandwiches this compound, PHF5A. So again, here's the wild type sequence, and you can see that in this case, you just get, that's the dominant sequence. What the guide does is give you a smattering, as you can see in this pale blue, of other sequences. But what's enriched when you add the drug is actually a 2 amino acid insertion that duplicates this threonine leucine dipeptide. So you get TL, TL in sequence. And that's what gets enriched over time. This Cas9 base editor actually has a nickase activity, and there's a little bit of a direct repeat in the DNA that we think can cause some slippage during repair that gives you, at a very low frequency, these sort of off-target or unpredicted changes in the DNA sequence, but they can be enriched by the compound. And so you can infer then that this is actually the causal mutation that's producing the resistance to the compound. Okay, so by doing a lot of this amplicon sequencing, one can then, again, map the mutations to structures. So what you can see here is where that threonine 1080 isoleucine mutation is. And it's actually on the alpha helix opposite that actually packs against the compound, but it's not directly, it's a residue that's not thought to be directly contacting the compound. So you have to remember these alleles have to be viable in the absence of drug, but have some effect on compound association-dissociation rates or conformational changes downstream of compound binding that allow you to get resistance. And this is the only mutation we got in SF3B1. We got many mutations in the other side of the sandwich in PHF5A, again, not in residues that directly pack up against the compound, but in sort of one shell away that in some way affects action of the compound. So we were happy to see this, and it was great. So we have a proof of principle experiment, but then the question was, so what? Do we move on to another screen that's more interesting and more specific? Well, we didn't immediately because we found these other proteins that they're guides against other proteins that produced resistance to the compound. And this protein, SUGP1, got our attention for a number of reasons. One is that there's no yeast ortholog, so it was never studied in yeast and can't be studied in yeast because it doesn't exist in Cerevisiae. The second was that this is was recently identified as a new tumor suppressor. And so this paper from Jim Manley's lab, the title says it all. The model is that disease-causing mutations in SF3B1, these are the cancer mutations, alter splicing by disrupting interaction with SUGP1. So what this paper proposes is that the mutations in human tumors in SF3B1 are oncogenic because they disrupt the interaction between SF3B1 and this mysterious protein SUGP1. The evidence for this, the initial evidence for this was a mass spec experiment where Manley's group tagged SF3B1 and expressed either the wild type SF3B1 or a tumor hotspot mutation in SF3B1. And then he did mass spec and compared peptide abundance, comparing wild type and this hotspot mutation. And for most proteins, there was no difference in the level of the proteins detected in the mass spec experiment. But for this protein, SUGP1, there was less in the tumor mutant complexes. And this was followed up by a number of more directed experiments. And the data in the paper are consistent with the model that these various different SF3B mutants that are found in tumors show much lower binding of SUGP1 to SF3B1 complexes. Further evidence that SUGP1 is connected to SF3B1 in cancer came from studies of the Cancer Genome Atlas, in which RNA-seq data was analyzed along with DNA sequencing data. So SF3B1 tumors produce changes in splicing, in particular the branch point that gets used tends to be, you tend to activate these cryptic upstream branch points and 3' splice sites. And so if you then use that RNA-seq data to find just the RNA-seq signature to predict the DNA sequence changes, you find mutations in SF3B1 in those heat repeats I mentioned before, but you also find a class of tumors that do not have changes in SF3B1 that have the same splicing changes, or at least similar splicing changes. And they end up being in this protein SUGP1. So these are SUGP1 mutated tumors that have substitutions all over the protein that again mimic the splicing phenotypes of SF3B1 tumors, further supporting the contention that SUGP1 and SF3B1 are connected when it comes to tumor formation. So despite the importance of this protein or the interest in this protein, there's no function known of this protein. And again, there's no yeast ortholog. The protein has some domains, including a domain called GPatch. And the reason I mentioned that is that when we sequenced our drug-resistant or compound-resistant mutants in SUGP1, they ended up being in or just upstream of the GPatch. So two different substitutions. One in the GPatch has a slightly stronger phenotype than the one upstream of it. These mutations cause changes in splicing, even in the absence of drug. They're subtle, so not too many genes are affected, but you can see them and validate them. So here's an example where in wild type, you only get this 3' splice site being used, but in the SUGP1 mutants, you activate an upstream 3' splice site. This is RNA-seq data. I never believe RNA-seq data unless it's validated independently with PCR, RT-PCR and gels, and we can see that this is one we could validate quite nicely. At the same time, we could do global RNA-seq, and these mutants on their own don't have global changes in transcript levels. They're quite subtle. They're not killing the cells or making them super sick. So we think the effects are likely to be direct. I mentioned the GPatch again before because our mutants map to the GPatch. And so what is the GPatch? Well, GPatch stands for glycine-rich patch. It's just a motif. And again, our mutants are in, one of our mutants is in a conserved glycine, shown here. This is just an alignment of SUGP1 with other GPatch proteins. And again, the other one is upstream. So if you work on RNA, you should know that GPatch proteins function as activators of a class of ATPases that I've already mentioned called DEAH box ATPases. So there is one crystal structure of a G-patch protein bound to a DEAH box ATPase. This happens to be a G-patch protein involved in ribosomal RNA biogenesis. And you can see here that it sort of winds its way through the structure of the dead box or DEAH box ATPase, which in this case is actually an RNA helicase called DHX15 in humans. And in vitro, this G-patch will activate the helicase activity and the ATPase activity of this, ATPase. In fact, in the spliceosome, these are widespread. In the spliceosome, there are actually two known G-patch DEAH box pairs that function in the spliceosome. One of them I've mentioned before, that there's a step in which this SF3B that comes back again, which again is clamping down on the branch point and gets removed before the chemical steps is ejected. This pair is well studied, well established, first in yeast. These are the mammalian names. And even more important or more equally striking is another DEAH box protein G-patch pair that disassembles the spliceosome after catalysis, called PRP43 in yeast or DHX15 in mammals. So G-patches activate these ATPases. However, the ligand for SUGP1's G-patch was not known. So how do you find it? So we turn to the latest and greatest, everyone's favorite technique, which is proximity labeling. And so here, what we're doing is using bio-ID, so a low specificity biotin ligase, BurA, mutant in BurA fused to our protein. In this case, we used a couple different versions of the turbo ID setup. And the experiment is simple. You take HEC293 cells, you transfect them, and add biotin, and then isolate the biotinylated proteins. After some optimization, we got this to work nicely. What's shown here is the silver stain of a gel using SUGP1 minus or plus biotin. When you add biotin, you get a bunch of bands. When you do the streptavidin pull down, when you do mass spec, you get a big list of proteins, including lots of proteins involved in splicing, including DEAH box proteins. Of course, we're massively overexpressing stuff in HEC cells, so you get the kitchen sink. It's a giant mess. So I thought the next step should be perhaps integrate a single copy of these fusions, more physiological levels of expression, et cetera. But we thought, well, if we really believe in genetics, one way of finding the biologically relevant interaction was to use the genetics. So why don't we compare wild type and the mutants we got out of our screen to see if there are any proteins that bind differentially. So we repeated the experiment I just showed you, comparing again wild type versus mutant. We did it in triplicates. We did TMT mass spec, so quantitative mass spec, and asked what's differentially labeled in our constructs. And to my amazement, only a single protein came out as significantly decreased in its labeling in our mutants. So this is again, comparing that mutant in the G-patch itself to wild type. Not only did we only have a single protein, but it was a DEAH box protein, which is what G-patches are supposed to activate. And it was even more interesting to us was that it's DHX15, which is only known activity in splicing is to disassemble the spliceosome. And this also rang true for another reason, which was that people had identified DHX15 in early splicing complexes, but didn't have a function associated with it. To proximity labeling isn't the same as binding if you're a biochemist. Luckily, Irene's office mate when she was a graduate student in Switzerland was this fellow Michael, who had solved the only crystal structure of a DEAH box protein to a G-patch. And so she called him up and said, hey, would you like to collaborate? He said, sure. So he's a student in Stephanie Jonas's lab at ETH Zurich. And so they can express versions of this G-patch in E. coli, purify it as an MBP fusion. They have recombinant DHX15 from insect cells. This is a pull-down experiment. What you can see is that SUGP1 pulls down DHX15 but our mutants show decreased interaction. This is a constructed mutant based on a crystal structure, which shows even a stronger phenotype, but it's clear that our mutants reduce the interaction. We can also do the same experiment a slightly different way, taking advantage of the fact that G-patch proteins activate RNA binding by DEH box proteins. So this is a fluorescence anisotropy experiment where we ask what happens when you add SUG-P1 to DHX15 to its binding to RNA. In this case, it's poly-U, and you can see that when you add the protein, you get increased RNA binding and increased affinity, and our mutants are defective in that stimulation of binding. So we're pretty convinced that SUG-P1 is a direct ligand for the DHX15 ATPase that disassembles the spliceosome. And so this leads us to a model. And our model is very similar to that proofreading model that I described earlier for yeast by John Staley. So I'll take a little time to go through it. So the idea is that this drug, plodainolide B, slows down the formation of an effective A complex, we'll call it. Realize there are many steps in between here, here and here. And that the normal function of SUG-P1, which associates with these early complexes, is to just disassemble them, is to take them and tear them apart. And this can lead to various fates, including just disassembly of the spliceosome or other events happening. The idea then is very simple, is that in our mutant, which we've shown disrupts this interaction, the discard pathway is defective. And now, despite the fact that the drug is still there and slowing things down, these spliceosomes have nowhere else to go. They can't be discarded, and eventually some fraction of them get through the pathway, allowing the cell to survive in the presence of some concentration of the drug. We think this can also explain the aberrant splicing one sees in tumor cells, in mutants in SUG-P1 or SF3B. If those cells are defective in this discard pathway, that might allow substrates that are poor substrates, for example, if the spliceosome lands on the wrong branch point, to be used and not proofread out of the system. One piece of evidence that appeared while these studies were in progress was a nice study from Melissa Eureka's lab at UC Santa Cruz, where she did some clever experiments to examine the role of DEH box proteins early in spliceosome assembly. And one of the upshots of this of her work was showing that if you partially deplete DHX15 from a helinuclear extract competent for splicing, she observed an increase in the assembly of formation on a gel, of the native gel, of this early splicing complex I mentioned, the A complex. And so her conclusion, in fact, was that DHX15 disassembles early spliceosomes, and that this could be a fidelity mechanism. So certainly her thinking is very much in line with how we're currently thinking about the mechanism here. So for those of you who don't care about splicing, but do care about genetics, the bigger picture for me as a yeast geneticist is that it's now feasible to do program genetic screens of essential machines in human cells and get the type of informative alleles that you could only get easily before in Saccharomyces. And these informative alleles can then be used, combined with biochemical experiments to learn something about the system. And so, and as I've also shown, just by, in our case, somewhat dumb luck, you can end up falling into a human protein that's involved in a human disease. So I think there are many opportunities now to ask questions that couldn't be asked in human cells about essential processes using the awesome power of yeast genetics. So I'm going to end there, and I want to, of course, thank Irene Bush, who's an amazing postdoc who drove every aspect of this project, and Beto and Susan were also instrumental in this project. These are the other members of the lab working on other stuff that I didn't have time to talk about. And have to thank Al Burlingame at UCSF and his postdoc, Juan Osez Prieto, who did the TMT mass spec Importantly, Michael and Stephanie were crucial for the biochemistry. We got advice from a number of people, but technical advice on working with these haploid cells came from Jan Corret at Stanford, who was very helpful in helping us with the ins and outs of this cell line. And of course, none of this would be possible without support from the NIH. And I'm ending here again with another pandemic snapshot. One of my routes around the city takes me by the bridge. And so this was a particularly nice day. So I'll stop there and I'd be happy to answer questions. Either Peter or Jason.
Yeah, I was just wondering, was DHX15 part of your screen, and would a hypomorph that would affect the ATPase activity be similar to the sub-P1 interaction?
Right. So the question is, should we have gotten DHX15 as a resistance mutant? In fact, we got DHX15 as a hypersensitive mutant. We don't know why, because DHX15 has other roles in splicing. The challenge of getting a mutation in DHX15 that would, for example, disrupt the interaction with SUG-P1 is that all G-patch proteins bind to the same substrate and has other essential functions in the cell. So it'd be very hard to get a mutation that it would be impossible, I think, to get a mutation that only affected the interaction with SUG-P1. It's also involved in ribosomal processing, so I suspect such mutants might just be dead. The other, of course, issue is that base editing only hits a certain fraction of nucleotides. So it's entirely possible that with a PAM-less Cas9, who knows if that would work, or an adenine base editor, might give us such a allele. So you can't conclude anything by what you don't get, unfortunately, but would have been nice. Jason?
Yeah, so I thought it was a beautiful talk, and I'm pretty excited about this strategy. And I think, when you saw on your slide where you saw the synthetic interaction between the compound and these alleles, you were surprised by that, but then you said, oh, well, I should have been surprised. But I'm actually still a little surprised because it seems like when you get these synthetic lethal interactions, it suggests that you have a really high editing efficiency in these cells to even see them.
Yes.
I would just naively think that you would mostly be able to select for positive.
Yeah, so it's always the case with CRISPR that if editing efficiency isn't great, it's mostly positive, right? For some reasons, and I think, again, sometimes it's better to be lucky than smart, base editing works better in these haploid cells than other cell lines. I don't know why, but I'm happy that that's the case. So that was another reason to be surprised at getting dropouts. And so, yeah, I got them. It works.
But you must have an estimation from the sequencing about roughly how efficient it was potentially?
We have we don't, because of we don't know what the phenotypes are. We need to do that. We've done that with individual guides for regions of the genome we know cells don't care about, and it varies, of course, but it's pretty efficient in general. Of course, not all guides work, but the guides that do work well. So. Aaron?
Are the Plod B resistance mutations also resistant to spliceostatin?
We have not checked whether they're also resistant to spliceostatin. I guess they do slightly different things, right? That spliceostatin covalent.
They're structurally unrelated. So maybe they get selectivity.
Well, we did derive clonal cell lines for doing the splicing analysis, so we could look at that.
The other question I had is, you mentioned early on that one of the parts of the secret sauce to this was getting a base editor that was not toxic to the cells. And I'm just wondering if you could expand.
Sure. So, like with all things CRISPR, there's like an enormous variety of things that are out there. Yeah. And so at one point, David Liu came through town and says, oh, I've got this base editor that's like super dirty. It's perfect for what you want. It'll change A's, T, it'll mutate everything, right? And so you try to make a cell line with that, and you get you either don't get a cell line, or you get a cell line and the base editor isn't expressed because the cells are unhappy. We've done that. We did that with a few editors early on, and like the sort of the editor we used was a version from this guy, Luke Dow, who's a cancer guy at Cornell, who fixed some of the problems in the early base editors, like he stained he stained cells after putting the base editor in the cells, and the Cas9 was all in the cytoplasm. And so he put some more NLSs on there and fixed a bunch of other things, so it worked a lot better. And so he made a whole set of lentiviral constructs and got them from him. We got some advice from him. It was great. And one of the sort of early generation base editors gave us a stable cell line. We haven't tried, we haven't done anything. The adenine base editors seem to not work very well in our hands in terms of efficiency. And then we haven't tried like the ones that are PAM-less or PAM expanded, because then you worry even more about off-target effects. I worry about specificity. So this seems good enough. And if you're a yeast geneticist, you know that you can get a lot of things just using EMS mutagenesis. You don't have to hit every base in the genome to get the allele you want. So pretty good. Dave?
Have you thought about doing a real yeast type selection where you see if you can take a reporter construct?
Yes.
Slice that you and see if you
Yes.
you get.
Yeah, absolutely. That's like aim 3 in the grant. So the grant's funded. So I just need somebody who wants to do that. Yeah. And Ben Blanco has made these cute reporters that if you include a microexon, you make one fluorescent proteins, and if you skip the exon, you get another. Yeah, I mean, there's like, I think there's a whole world out there, right, to explore. Not only for splicing regulation, but I'm also interested in transcription splicing coupling, which is a super murky area. And I think there's lots to do there. So I think it's kid in a candy store time. It's not as fast as yeast. You can't make a clonal cell line by just streaking cells on a plate, waiting 2 days, say, hey, I got a clonal cell line. Instead it's like a month or two, but if you're willing to put up with that, you can do stuff. Question in the back, I can't, this lights are.
A couple of times that the sequence space that's accessible
Yeah.
to editors is very small.
Yeah.
It would seem to be that the utility here is that it's a screen for identifying regions and parts of the U2 snark to go after in a more deeply mutagenic fashion. And so now that you have these EHAP cell lines that are pseudo-stably haploid, I wonder if you're going to try a saturation genome editing specifically on the regions that came out of your initial base editing.
I mean, Jay Shindur has done that with BRCA1, right, a few years ago. But those are kind of painful because the efficiency is not great. But HDR type efficiency is not great, so they're laborious. You have to clone out 96 little cell lines to find one HDR event typically. So it's sufficiently painful that I wouldn't propose it to anybody in the lab. If there was a better tool, like maybe one-day prime editing will be efficient enough that one could tile a larger region. That hasn't so far been developed to a point where it's like a super efficient tool. It'd be fine for positive selections for sure, yeah, so you have to, because everything is slower than yeast or any fungal species, one has to pick one's battles carefully because everything seems like months of, prep time to do an experiment. That's why I still like yeast.
There are any more questions? Thank you again.
Thank you.